Hello, everybody. Hope you're doing well today. Today we're going to look at Chapter 15 and the chapter questions. We're going to run through those together. This is on pages 312 and 313 in your textbooks. If you want to follow along in your text, you can use that to help you solve these problems. All right. So if we look at our first question, it is question number 1. It begins, a man with hemophilia, a recessive sex-linked condition, has a daughter without the condition. Okay, so I strongly recommend always begin by writing down the things you know. So it's X-linked and it's recessive to have hemophilia. So let's say an X with a big H means the person will be, let's say, healthy, and an X with a little h means the person will have hemophilia. Now, when we say something is X-linked, that means the gene is found on the X chromosome. And since females and males have different numbers of X chromosomes, we need to keep track of the gene on the X chromosome. So if they tell you something is autosomal, you can just use your like big A, little a, big B, little b, all that business. But if they tell you something is X-linked or sex-linked, then we need to keep track of that gene on the X chromosome. So we write the X chromosomes and we put the genotype in superscript. So we know that he's a man, and we know that he has hemophilia. So we'll put down his sex chromosomes, XY. He has hemophilia, so he has a little X. Let's see. Said he has a daughter without hemophilia. So we know the daughter's sex chromosomes. They're XX. She doesn't have hemophilia, so we know she has a dominant allele. What's the only thing her dad could pass on to her? An X with a recessive allele. So that's the daughter's genotype. So she marries a man who does not have hemophilia. So let's say here's the hubby. Here are the hubby's sex chromosomes. He doesn't have hemophilia, so he'll have a dominant allele. They want to know the probability that their daughter will have hemophilia. Okay, well, we can set up a Punnett square. And we'll put the husband's genotype, the daughter's genotype. And then my recommendation is sort of filling out the X's and Y's, and then adding the alleles in superscript. Okay, so the first question, what is the probability that their daughter will have hemophilia? So we'll say odds that the daughter will have hemophilia. Well, if dad doesn't have hemophilia, he's only going to pass on dominant allele. So the odds the daughter would have it, 0, no chance. The next question is, what about a son? Okay, so the odds that a son would have hemophilia. Well, if we look at the son, we're looking at just the boys, so that is 1 out of 2. So odds are half. And then the final question here, if they have 4 sons, what's the probability that they'll all be affected? So we want to know odds, all 4 sons. So the odds of the first son would be a half. Odds of the second son, still a half. Odds of the third son, hey, he's his own independent event. That's a half. And odds of the fourth son, it's a half. So 2 times 2 is 4, times 8 is, times 2 times 2 is 4, times 2 is 8, times 2 is 16. So the odds are 1 in 16 that if they had all 4 boys, that all 4 of them would have hemophilia. So you have to be very careful with the question they ask, because if they said, what are the odds that their daughter would have hemophilia? We look just at the girls. If they say, what are the odds that their son would have hemophilia? They look just at the boys. But then if we look at what are the odds they'd have a child with hemophilia, then the answer would be 1 out of 4. So again, pay close attention to what they're asking you. Because if they say, what are the odds that their daughter will have something? You look just at the girls. What are their odds their son will have something? You look just at the boys. And if they say, what are the odds their child will have something? You're going to look at everybody. And that alters the odds. All right. Our next question is number 2. What's number 2 getting at here? It says pseudohypertrophic muscular dystrophy is an inherited disorder that causes gradual deterioration of the muscles. It is seen almost exclusively in boys. So that's a key idea here. It happens in boys. Born to apparently unaffected parents. So that's another key idea here. Unaffected parents. Let's see. And usually results in death in the early teens. That's rough. Is the disorder caused by a dominant or recessive allele? Well, if the parents don't have it but the kid has the condition, it's likely that it is a recessive condition. Let's see. Is this inheritance sex-linked or autosomal? Well, since it's affecting males and females differently, it is likely that it is sex-linked or X-linked. Let's see. How do you know? Explain why the disorder is almost never seen in girls. Okay, so a couple things to think about. If something is sex-linked and recessive, that means to have the condition, you have to have 2 recessive alleles. So since males only have 1 X chromosome, if they inherit the recessive allele, they've got the condition. Females have 2 X chromosomes, so that in essence serves as a backup. Now, because females have 2 copies of the X chromosome, they get a second chance to inherit a dominant allele, making it less likely that they'll have sexually recessive conditions. Additionally, they made the point that males die in their teens. So if they die in their teens, they're probably not passing that on very frequently. And the only way a daughter would have the condition is if both her mom and her dad have the condition. And if males are dying in their teens, they're probably not passing it on to their daughters. So it's going to be exceedingly rare for females to be affected by something that is sex-linked recessive and kills males early on in life. All right. So on that note, let's move on to something else. Okay. Our next problem is number 3. So number 3, a wild type fruit fly. Okay, this is getting into the Thomas Hunt Morgan fruit fly experiments. So in the Hunt Morgan fruit fly experiments, let's maybe take a quick peek back here. They were doing matings with these fruit flies, and what they were doing is mating like homozygous dominant flies with homozygous recessive flies and creating these heterozygous offspring, and then doing what's called a test cross with homozygous recessive offspring. You can imagine, back in the early 1900s, they couldn't do genetic testing on organisms they look at. All they could do is look at traits. So if you see something that looks dominant, you want to know, is this homozygous dominant or is it heterozygous? Well, what you do is perform what's called a test cross. With a test cross, you take an organism with an unknown genotype that displays dominant characteristics and mate it with one that has a known genotype that's homozygous recessive. Now, if these genes were on separate chromosomes and sorted independently, you would predict there would be a 1 to 1 to 1 to 1. Or I'm sorry, you'd expect that all the offspring would look, if they are on different chromosomes, you'd have equal odds of every possibility. Okay, 1 to 1 to 1 to 1 ratio. Okay, if they are on the same chromosome, then the gene should always look like one parent or the other. Now, in the Hunt Morgan fruit fly experiment, they didn't get either one of those things. Most of the offspring looked like one parent or the other, but occasionally the offspring would be a unique combination. So something seemed off because the offspring weren't having equal probability, showing that the genes are on different chromosomes. But somehow the information was getting swapped around. So that's where they figured out something called crossing over is occurring. Okay, so this is sort of our first understanding of crossing over. So let's look at number 3 to sort of see how they figured this stuff out. Okay. So it reads, a wild type fruit fly. Again, wild type is what you typically see in nature, and it's usually the dominant condition. So a wild type fruit fly, heterozygous for gray body color and normal wings. Again, I'm always in favor of writing down what you know, even if it takes a little time. So if you have a dominant condition, you have gray body color. If you have the recessive allele, that is black body color. So heterozygous for gray, and then again, they give this term normal wings. So we use capital N for normal, and a lowercase m for vestigial wings. That means they have these little dinky wings that they can't use for flight. All right. So the fly is heterozygous for these traits. So that's big G, little g, big N, little n. Let's see. It is mated with a black fly with vestigial wings. So that means it's homozygous recessive for both traits. Again, this is a test cross. Okay, so what do we get? The offspring have the following phenotypic distribution. Okay, so 778 were wild type. Wild type is again, what you typically see in nature. It is the gray normal. And there were 778 of those flies. That's counting a lot of flies. Let's see. Black vestigial, that's the other parent. There were 785 of those flies. Okay. Let's see. Black normal wings. There were 158 of those flies. And then finally, gray body with vestigial wings. There were 162 of those flies. Okay, so in total, there were 1,883. What a year! 1,883 flies. So the question is something. What's the recombination frequency between these genes for body color and wing size? So again, if the genes are located on the same chromosome and always inherited together, the offspring should all look like one parent or the other. And if the genes are on separate chromosomes and you have independent assortment, you should have equal probabilities of all these. But what's happening is the genes are usually inherited together. The genes from the parents are usually inherited together, but sometimes you get a unique combination. So that means sometimes crossing over occurs. So what we're going to do is calculate this recombination frequency. And the way you do that, you take the total number of offspring that are, this is key, unlike the parents. This is a bad attempt at an R. Okay, so it's the total number of offspring unlike their parents, and you divide that by just the total number of offspring. So again, how often is recombination happening? How often are these genes getting swapped around between chromosomes? It's the number of offspring that are unlike the parents divided by the total number of offspring. So in our instance, there are 320 offspring that are unlike the parents. Divide that by your 1883, and that gives us a recombination frequency of 17%. So that means 17% of the time these genes for body color and wing shape get swapped during crossing over. If you look at the Hunt Morgan data, it was 391 flies out of 2,300. Turns out also 17%. So our experimental numbers match up the Hunt Morgan experimental numbers, and boom, we're pretty happy with that. Okay, so let's see if we can, excuse me for just a moment. I just realized it'd be helpful to draw something to try and help us visualize what's going on with this stuff. So let me sort of imagine what we've got going on. If you have, like up here, we have this heterozygous parent. So we have a heterozygous parent that is heterozygous for these two genes. Okay, so here are the chromosomes. Let's see, they're gray and normal. So this parent is gray and normal winged, and the other parent is black vestigial, so that's homozygous recessive. So that is a little g and a little n. Now we know before meiosis can happen, you're going to copy that DNA, right? So you know what? Let me do this in a different color so it makes these stand out a little better. My apologies. Okay, so this DNA is going to get replicated just to make things a little more visually complex. Let's see. So again, let's do this. We've got the replicated DNA. I'll make my X's differently, or a little bigger next time, so I've got more room. That's bunk. Okay, so again, here is a chromosome made of two sister chromatids from the parent who's gray normal. Here is the chromosome with the parent who's got black-bodied vestigial wings. Now, if you have no crossing over and no recombination, same thing, no crossing over, no recombination, then the offspring that result from this are going to get gametes that are either dominant for both traits or recessive for both traits. Okay, so these offspring would be what? Gray and normal. And these offspring would be black vestigial. Okay, so again, if you have a parent that is heterozygous, they get a big G, big N, little g, little n. You don't get recombination. That's all they're going to pass on. Right? Now, let's imagine a scenario where crossing over will occur. So again, you start off with your heterozygous parent. I'll space this out a little better this time. So we have what we had originally. The DNA has been replicated. little g, little g. And if crossing over happens, let's label this. Then, we got our crossover or recombination, again, however you want to phrase it. The gametes that result from this, we're going to have a little more variety here. So in some instances, crossing over wouldn't have affected certain sister chromatids. So you still have your big G, big N scenario. But if crossing over does occur, you could have a little g and a big N. Or you could have a big G and a little n. Or again, certain chromatids may be unaffected, so you have the recessive condition. Okay? So what crossing over does is it mixes up these genes in the parent so that the gametes that are produced have more variety. And that's what is causing this recombination to occur in the Hunt Morgan fruit flies. All right, let's move on to number something else. Number 4. Number 4. Ooh, lots of words. A planet is inhabited by creatures that reproduce with the same hereditary patterns as seen in humans. That's helpful. Three phenotypic characters: height, head appendages, and nose morphology. Okay, so let's record what these traits are. Again, I think it's always helpful to sort of see the pieces of the puzzle when we try to solve it. So tall is dominant to dwarfism. Goodness, can't write R's. In terms of the head appendages, dominant is having antennae, and recessive means no antennae. How sad. In terms of nose morphology or nose shape, the dominant allele means you have an upturned snout. I'm sure it's lovely. And recessive means you have a downturned snout. Somehow neither sounds great. Now, if letters are going to look similar in capital or lowercase, like in the case of S, for the recessive, I tend to just write the letter in cursive. Again, some people put like dots or lines, whatever works for you, as long as you're consistent and able to observe stuff. Okay, so it says, since the creatures are not, quote, intelligent. Oh no, this is traveling down a path I maybe feel a little uncomfortable with. Earth scientists are able to do some controlled breeding experiments using various heterozygotes in test crosses. Okay, so we have heterozygous things doing test crosses. All right. Well, our first scenario, we have tall heterozygotes with antennae. So this is our first scenario, A. They're tall and heterozygous with antennae. And they said it's a test cross, so then you use homozygous recessive. Let's see. What do we got going on here? It says the offspring are tall with antennae. And that number is 46. We have dwarf with antennae. That number is 7. There is dwarf with no antennae, which is 42. And finally, we have tall with no antennae, and that is 5. Now, the parents were tall with antennae, so put a little asterisk there, and dwarf with no antennae. So let's just make a little mark here, a little note to ourselves. Say this is like parents. Okay. So they want us to calculate the recombination frequency. Okay, so recombination frequency, again, we said it is the number that are unlike their parents divided by total number of offspring. So this ends up being 12 that are unlike their parents, and it is 88 plus 12. It's 100 offspring total. So the recombination frequency is 12% for tall, the gene for height and antennae. All right, so that's the first part. Part B, read something else. Antennae. Oh, for heterozygotes with antennae and an upturned snout. So heterozygous with antennae and upturned snout. So heterozygous for antennae, heterozygous for upturned snout. Again, it's a test cross, so the other parent is homozygous recessive. Okay, the offspring are antennae with upturned snout. That is like one of the parents here. There are 47 of those offspring. Let's see. There are individuals with antennae and a downturned snout. There are 2 of those. There are individuals with no antennae and an upturned snout. Let's see here. I'll make sure I'm counting this correctly. 47 antennae and a downturned snout. Wait a second. I'm sorry, I'm getting this messed up here. Offspring antennae and an upturned snout is 47. Thank you. Antennae and a downturned snout is 2. No antennae and a downturned snout. Okay, that's 48. And then the last option is no antennae and an upturned snout. That is 3. Okay, so the parents are antennae and upturned snout and no antennae and downturned snout. So the parents there. So to get our frequency, we just take the number that are unlike the parents, which is 5, divided by the total number. Again, it's still 100. So the recombination frequency in this instance, 5%. So 5% of the time, the offspring are unlike their parents. All right. Let's do, well, it's been about 30 minutes. Let's give you a quick break. We'll pause the video here, and then we'll finish up the last question.